In this video lecture, I will be talking on antigens. An antigen is a substance that capable of inducing a specific immune response, commonly are referred to as antigens, more appropriately called an immunogen. So, immunogen is a substance that induces a specific immune response, humoral or cell-mediated immunity. For example, B cell interact with B cell antigen or B cell or antigen displayed on the surface of B cell in association with major histocompatibility class 2 interacts with TCR of T helper cell, and this will lead to activation and proliferation of and differentiation of B lymphocytes into plasma cell and memory B cells. Also, other examples: TCR of CD8 interacts with antigens presented or displayed in association with major histocompatibility class 1 on surface of target cell, and this will lead to differentiation of T cytotoxic into cytotoxic T lymphocytes and memory T cell. However, substances which are not capable of inducing immune response or responses are known as haptens. Haptens are a small molecular weight. They have an antigenicity property, but they are not an immunogen. So now I am going to talk about types of antigens. I will start by giving just a short account on Thymus-dependent and Thymus-independent antigens. Thymus-dependent antigens, this requires T and B interaction, and the antigen is usually a protein, and as can be seen here, this is T helper 1, which usually interact with antigen in association with major histocompatibility on the surface of antigen presenting cell. This, for example, macrophage. However, T helper 2 interact with antigen expressed on the surface in association with major histocompatibility in the surface of B lymphocyte in association with major histocompatibility class 2. So this is known as cross speak or cross talk. Usually T helper 1 talks with macrophage. T helper 2 like to speak or to talk to B lymphocytes, and actually these antigens are usually they are protein which has no repetitive epitopes in general. However, in thymus-dependent antigen, as can be seen here, the antigen is usually is composed of repetitive identical epitopes on surface of some, this is a microbe, and cross-link BCR, and this lead to independent activation of B cells. Activated B cell produces antibodies But however, these antibodies are weak, no class switch, no memory cell. And this is to say that B cell differentiate into plasma cells. However, this cross-linking provides with the first signal. The second signal is required by toll-like receptor on the surface of B lymphocyte, and these receptors actually interact with pathogen-associated molecular patterns on the surface of bacteria, and this provides with a second signal. So repetitive identical epitopes on surface of some microbe cross-link PCR and lead to independent activation, and this is the first signal. The second signal of B cell activation is generated by toll receptors on surface of B cell that interact with pathogen associated molecular patterns on surface of the microbe. Other classification of antigens, and it is according to the site whether they are exogenous or endogenous. Exogenous antigens, they enter body from outside, for example, bacteria, viruses, fungi, parasites, etc. Endogenous antigens, these are produced inside the cell. Usually these are viral and tumor antigens produced by or inside target cells, virally infected cell, and tumor or cancerous cell. Also, there is another type of antigens known as autoantigens. And auto, it means self. These are self antigens. But despite that they are self, they are not, I mean that they are not foreign, they become target for the immune system. An example of autoantigens, sperm and cornea. As you know that sperm and cornea are sequestered antigens, but when seen, the immune system attacks them and produce humoral and cellular immune responses. However, they should not be seen for life. But however, when seen incidentally, for example, in case of infection, inflammation, or trauma, so the immune system will recognize them as non-self. Also, in case of netosis, these are the neutrophils. The neutrophil has a capacity to produce some filaments, and following, for example, infection, these filaments actually they modify their chromatin and they produce these sticky filaments. However, some antigens will come outside due to this process, for example, proteinase 3, myeloperoxidase, and this will be seen as non-self, so the immune system will produce immune responses against them. So these antigens are hidden, so exposure of hidden antigens, as that occurs during netosis. Neoantigen formation, and this is due to gene mutation. For example, tumor antigens, these are self antigens, but undergo some alteration, for example, mutation, and when expressed on the surface of tumor cell, they will be seen as non-self. Tumor cells or tumor antigens are self-altered antigens. So they are self in origin, but they undergo some alteration, and due to that, they will be seen as non-self. And these antigens actually present on surface of tumor cells or secreted in bloodstream. For example, tumor-specific antigens. Tumor-specific antigens, these are specific to ionic cancer. So, according to that, classes of tumor antigen, there are two types or two classes: tumor-associated antigens, and these are expressed at lower levels of healthy cells. But however, tumor-specific antigens are found only in cancerous cells. Thank you very much, Professor Ahmed.